All right, so I'll go back just a touch. This is where I left off last time. There it goes. Yes. Yeah, I forgot to do. I'll do that right after this. Sorry about that. All right, so here's where we're going to come back to over and over in this class. Like I said last time, so the central dogma is originally proposed by Watson and Crick, includes information transfer from the storage of DNA into RNA, which is the intermediate that is then translated into protein. And going from DNA to RNA is easy because it's just base pairing. Still, it's instead of A's, T's, G's, and C's, it's A's, U's, G's, and C's in RNA. And then, as we'll see, the trick comes in going from the 4 letters of DNA into the 20 letters of protein. And that's where we're going to have transfer RNAs come in that are going to serve as an adapter for that purpose. So there are multiple levels, as we talked about last time, of regulation. So transcription is controlled, as I said, both by whether transcription factors or proteins are present that are necessary to turn on each gene by interacting with the promoter, and also, as we're going to talk about today, how the DNA is packed. So a lot of the DNA, even when it's not in chromosome form, is packaged so tightly with the histones that the transcription factors can't get in there anyway. And that's important because it allows another level of control. So if all the DNA was accessible all the time, you would need a ton more transcription factors to do that. We don't have that many. There are a lot, but that would be even more. As we'll see coming up, translation is often regulated, so you can get messenger RNAs accumulating, and then they can be held off on translating into protein until later on. Protein modifications like phosphorylation will help to turn existing proteins on and off, so you can have proteins sitting there, turn them on and off. So cyclins that control the cell cycle that we don't have time to talk about here, but you will in cell biology, so register for that in the spring. They are phosphorylated and dephosphorylated to manage progression through the cell cycle. So G1, S phase, G2, and then into M phase, whether that's meiosis or mitosis. And then more recently, I mentioned last time that the phenomenon of RNA interference. So RNA interference originally evolved as a defense mechanism against viruses that have RNA genomes, but now it has gone on and has been co-opted as a general regulatory mechanism. So changes to the central dogma. First, there's a fly on the screen. The first major one was when a researcher named Howard Temin, who was, and I think still is, I don't think he's dead yet, at the University of Wisconsin at Madison, discovered an enzyme present in many retroviruses that will allow RNA to be used as a template and may allow you to make DNA based on that. So previously it was thought that DNA was always the template, and you could make a DNA copy from DNA, or you could make an RNA copy, and this was the first example of it going back the other way. I'll get just a second. And we'll see in chapter 12 coming up, which I just posted the slides for, when we talk about DNA replication, maintenance of the telomeres, those sequences on the ends of the chromosomes, actually require the action of a reverse transcriptase, so maintaining their length. the template that's used to elongate telomeres is actually an RNA molecule contained within another molecule called telomerase that we'll get to then. Yes? So when you're talking about genes on DNA, and I didn't hook up my computer yet today, or my iPad, there's 2 strands of DNA, right? You can have a gene on either strand, but generally, except for some virus examples, there's never going to be a gene on both strands because of the way transcription happens. So we're talking about positive RNA and negative RNA, right? So plus strand or positive strand RNA is RNA that will actually directly code for proteins. Negative strand RNA is the complement of that, the other bases. And while I talk, I'm going to hook this up so that I can actually tell you about it. So you heard about this in microbiology class. All right. So we've talked about bacteriophage. They have their genomes are always made out of DNA, and then human viruses can have genomes made out of either single-stranded or double-stranded DNA and either single-stranded or double-stranded RNA. And if the RNA is single-stranded, then sometimes it's the plus strand, which is the strand that actually could code for protein, and sometimes it's the negative strand. But either way, it doesn't matter because almost always that RNA will be converted into DNA and the virus can go into the genome that way. The difference is, if I can get this fired up, the difference is that plus strand RNA, when the virus fuses with the cell, can be directly translated and get everything going right away. So let me try to flip this over now. So the question was, what's the difference between plus strand and minus strand RNA? Let's start out with DNA. So here's DNA. And we'll talk more about genes when we talk about transcription and also RNA processing and translation. But generally, you can have a gene where this part that I've circled, 5' to 3', will code for a protein when it's translated into or it's transcribed into RNA. But in that case, this part isn't going to be a gene, right? Or let's leave that there. Or this part could be what is made into RNA. But we always draw it 5' at the left and 3' at the right. So this So you can see that either of the 2 strands of DNA, the top one up here or the bottom one down here, can code for a gene, but in our genome, in the genome of almost every other organism, you're never going to have, like, for example, another gene being coded for here, by that top strand. The reason is because when we talk about transcription, you have to open up the DNA and have RNA polymerases using that DNA as a template to make the RNA. And if you're having them, it's like having two trains on the same track bumping into each other. That's not going to work. The only exception that I'm aware of to this rule is that some DNA viruses will have the same areas code for more than one gene. And the reason is because of what we talked about earlier, that limitation in the size of the genome of bacteriophage or viruses is the size of the capsid. A cell has, so what encases a cell? What surrounds the cell and separates it from everything else? The plasma membrane, right? If you want to make a cell bigger to some extent, you just make more plasma membrane. It's just a phospholipid bilayer. You can make it bigger. The nucleus, nuclear envelope, is two phospholipid bilayers stuck together with some pores between them. Likewise, if you want to make the genome a little bit bigger, all you need to do is add more phospholipids and expand it, right? But the capsids on those viruses, remember I said, can assemble in vitro. Viruses are not living things, and bacteriophage are not living things. So there is a serious space limit inside that virus. It's like a piece of hard-sided luggage, right? No matter how hard you cram, there is an absolute maximum that you can get in a piece of hard-sided luggage because the sides won't bulge like on soft-sided luggage. And so viruses a lot of times have to do this down here and have something coded on both strands because they only have a certain amount of total base pairs to work with. The way that they make that work and get around it is that, for example, the green gene here will be used early on in the viral infection and replication process, whereas the blue gene up here is going to be used later on. So even in that case, you're never going to get simultaneous transcription because the two genes are never going to be on it temporally coincident, so at the same time. Okay, so when you use, let me do this. If this is the DNA that is actually going to be coding for protein, not the other strand, then that's going to be the plus strand and the bottom's going to be the minus strand. So as we'll see in transcription, if you want to make RNA that looks like what I've got circled in green here, you're going to have to use as a template this DNA down here to make RNA that looks like the green strand. That's actually going to code for the protein. So some viruses will have RNA genomes that are, if this is what DNA would look like, and the gene is going to be on the green strand, if the coding area is actually right here, then a plus-stranded virus would have RNA with the same sequence as this green DNA strand I've drawn, and negative RNA viruses would have that one, a strand of RNA that matches that. Some viruses have double-stranded RNA as their nucleic material or their genome, in which case you would have something that looks exactly like this, only it would be, RNA instead of DNA. Now, plus-stranded and minus-stranded viruses, in those cases, those viruses are probably, if you can say it's plus-stranded or minus-strand, it means that there's only going to be genes on one strand, I would imagine. All right? Does that help? All right. So, a digression, yes, but this is good because you're asking now, and you're going to see this twice because this will help when we talk about transcription later on. So viruses that have an RNA genome will need to do reverse transcription to make a DNA copy, and then that inserts in the genome. So retroviruses, the big one that everybody knows being HIV, does this. So the reason why you can get an HIV infection and then it'll be years before you get the syndrome or AIDS from that is because the HIV virus, which is an RNA virus, will make a DNA copy of itself and then park that in your genome somewhere. And you don't have any active viruses. So you're not making any antibodies to the HIV virus, and you will appear, to be healthy for, 5, 10 years. And then something will happen in your physiology where you will be under stress, and that will cause the virus genes that are parked in your DNA to be transcribed, and then the RNA that is part of the genome will be made, and also the capsid pieces will be made, and new virus will be produced. When we talk about RNA interference at the end, it was also discovered that RNA can be used as a template for production of more RNA. So RNA interference is going to be a defense mechanism against RNA viruses that, as I said, is used for other purposes now too. But when you get a small amount of RNA, if you want to raise the alarm and spread the signal to other cells, you replicate this to make a whole bunch of RNA that looks like the RNA that came in. That way you will amplify the signal and make it more powerful. All right. So nucleic acid secondary structure. The main secondary structure for DNA you already saw in the pictures on the screen and the model I brought in. It's that double helical structure. But within both DNA and RNA, you can also make some other secondary structures, and these are derived from inverted complement sequences. So if you start out here and you look over there, you're going to see as you go in from the end, there's a T and an A, there's a G and a C, a C and a G and a C, etc. And those will make, see the outer, inner, until you get to this part in the middle that's TAC, TCA, TAC, TCA. These can base pair to form what's called a stem loop structure. So in both DNA and RNA, this one is also called a hairpin because it sort of looks like a little bobby pin that you would clip your hair up with. So in addition to that, DNA is very flexible. So instead of stem loops, you can also make just stems only, all kinds of stuff like this. So these are involved in regulatory functions. So we talked about RNAs being able to be turned on or off for translation. You can get some kind of a big structure like this at the 5' end of a messenger RNA that will actually help determine whether it can be translated or not. So RNAs have been for a long time known to form these really elaborate secondary and tertiary structures. So in all these, in this big case here, here's a little picture. Everywhere where the strands are together, you will have complementary bases, like you can see. Oh, that's not good. These are not good. The artist screwed up. So these are all good. That one's no good. That one's no good. That one's no good. That one's no good. This one is good. I never noticed that on there before. So these should all be complementary structures. I'm going to have to make a note of that to complain to the publisher for when they put out the next version. They should all be complementary, and then like this sequence in here in the loops is not. This is the two-dimensional structure. Like if you took this RNA and flattened it out in a book. This is not what this structure would look like in real life. It's so you can see where the base pairing happens. In real life, this will pop up into a tertiary three-dimensional structure that will still have all these base pairings, but it isn't going to look like this. There are some RNAs that can even function as enzymes. And the first one of these to be discovered was a messenger RNA for a eukaryotic organism that was able to splice out its own introns. And once this was figured out, then RNAs as enzymes was seen as possible instead of impossible as it was before, and people were looking for other instances of that. And so two instances where we'll see this occurring is, again, in splicing. So there are some messenger RNAs that can self-splice, but even the messenger RNAs that require machinery to be spliced, the actual splicing reaction is catalyzed by an RNA, not a protein, which is pretty amazing. It's an RNA having a catalytic function. And the second example is during translation. So does anybody remember from introductory biology? So proteins are made out of what? Amino acids, right? And how are they put together? So DNA is put together with what kind of bonds? What kind of bonds link the sugars together? Covalent bonds, but what's the specific example? The phosphodiester bonds, so the phosphate with the oxygen on each side. So what is the bond type that makes amino acids into a protein polymer? Peptide bonds. Peptidoglycan is for bacterial cell walls. So it sounds, yeah, I know, it sounds the same. There's all kinds of words that are the same. So peptide bonds. Amazingly, after all this stuff came out, it was determined that, so what's supposed to put amino acids together to make proteins? What's the big organelle? The ribosomes, right? And so it was thought that the proteins were there, and everybody knew that there were ribosomal RNAs, but everybody thought they were just structural. It turns out that part of the 23S ribosomal RNA, the biggest one in the big subunit, that's actually what has the catalytic function for forming peptide bonds. So figuring out something simple led scientists to go on and do other stuff. So this led to further confirmation of the theory of RNA world. So as organisms were evolving, it is now thought that the first nucleic acid was actually RNA. So right now we have DNA as being the storehouse of information. Then you go to RNA, which is relaying that information, and proteins, which are going to do the jobs in the cell. But previously, RNA had to function in all of those capacities, the storage of the information and also the catalytic functions. And finding all these RNAs that can fold up into three-dimensional structures and act like enzymes, so they're called ribozymes for that reason, is evidence for that. All right. So we'll talk about this a lot more when we get to the transcription, not transcription, but transcriptional regulation way at the end. In addition to DNA being available or unavailable for interaction with transcription factors, there also can be modifications made to DNA that can change its transcription status. And one of those is methylation. So this is a cytosine. Just replacing a hydrogen with a methyl group there will, in eukaryotes, mark this for transcriptional silencing. So protein enzymes will recognize cytosine methylation and then we'll come in and shut down that area so that transcription doesn't happen. This is another example of epigenetics we talked about earlier, and it's actually the mechanism for it. Remember the immunoglobulin-like gene that's turned on in the male genome and off in the female genome? Well, I don't know if this is exactly the mechanism, but methylation is one of the mechanisms that could cause this gene to be turned off in the female genome. So when those genes are reset in the germline, in males, both chromosomes are set to be unmethylated, and in females, both of them would be methylated so that that gene would be turned off then. In bacteria, methylation can mark DNA as foreign, and in that case it will be chopped up, versus self-DNA, which is immune from this process and therefore spared. So in eukaryotes, cytosine methylation is always in CpG pairs. So what is a CpG? It's just a cytosine, the P is just the phosphate, and G. So a CG doublet. Why is this? Because so why is methylation in eukaryotes always in CpG pairs? So you have a CpG like that. The reason is because of complementary base pairing. If you have a C and then a G, 5' to 3' on one strand, you will have to have a C and a G, 5' to 3' on the other strand. Because these strands have to separate for DNA replication, as we'll get to in chapter 12, it is helpful to have both strands methylated, and by having a CG dinucleotide, both strands have to be methylated there. So when these two strands separate for DNA replication, new bases will be added to them, and then you get this. So here's going to be the new DNA. For the top strand, and then the bottom strand is going to look like this. And you can see that you end up with what's called hemimethylation. So this strand, the original template strand, is going to be methylated, but this one down here isn't, right? New DNA synthesis. And now enzymes can come in and see that you have a methyl here, and then go add a new methyl there, and likewise for the other strand. That's why you need to have both strands methylated if you're going to be able to propagate that mark down through the generations. Okay, so that's it for chapter 10. We're going to move on to chapter 11. So chapter 10 is the structure of DNA and RNA. DNA is actually packed into the nucleus of eukaryotes, or the nucleoid area of prokaryotes. All right, so the figure in the book is just this outline around here, but I've put some exciting examples of packing things into small places. They used to have these things called phone booths that you may or may not have seen. If you've never seen a phone booth, you should watch Bill and Ted's Excellent Adventure, because, you know, it's a top movie pick. Or the modern version, like jamming people on top of cars and trains and so forth, trying to package a lot into a little area. So the green outline border around this slide is to scale the size of the E. coli genome. It's circular. Up here, to scale with the DNA, is the size of the actual E. coli bacterium. So that is a lot of DNA to stuff into a very tiny area. How much DNA do you guys know? Do they tell you in other classes the length if you took all of your chromosomes and held them out, end to end? What's a guess for how long that would be in the metric system? What's a guess? Say something that you think is too big. What? A what? A billion what? Your genome is 3.02 billion base pairs. I'm talking about how long would it be? A billion feet. No, that's a little bit much. No. All right, so never mind. I've got you thinking too big. It is 2 meters. A disappointment. Wow. I guess I shouldn't build it up. But each of your cells has 6 feet of DNA in it. You got to be thinking scale-wise, right? If you had a mile of DNA and you crammed it all together as much as you could, there's no way that it would fit into the volume of a cell. And you would think that this wouldn't be able to fit either, but it does. So how does that work? The first way that happens is through a phenomenon called supercoiling. And shoot, I forgot to bring my rubber hose with me today to demonstrate this, but I'll bring it next time. It's a fabulous demonstration. Actually, it really does work exactly like shown here. So what is supercoiling? What is regular coiling? If you have a circular piece of DNA that is linked at both ends, DNA wants to be about 10.5 bases for every 360-degree turn, right? And I showed you that in the DNA molecule. So bDNA is about 10.5 bases per turn around the helix. We're just going to say 10 here to show that. So bDNA is 10, aDNA is 11, and what's in cells is about 10 and a half. So that means if this DNA has 360 bases, there will be 36 full turns around the helix, 1 turn for every 10 bases. Because it's closed circular DNA, that means here's one strand of DNA, here's the other strand. It is closed like this. It's locked around, so it can't move. It can't twist more or twist less. Here's an electron micrograph of a piece of DNA like that. Another anachronistic example, but you might be able to find one around, is a phone, a landline phone. Remember those? They still have them here, like in offices and stuff. You ever seen a phone cord that looks like that? But is it really hanging like this in a perfect circle? No. What's it look like? It's all twisted up because it's been turned around a bunch of times. So in this case, even though it's a single helix and not a double helix, the twisting of the phone cord is like the turning of the DNA around the helix. And it's closed because the phone is sort of like on the hook here. If you would take the phone and lift it off the receiver and turn it around once and put it back on, you would be either adding or releasing a turn into this helical structure. But just like the rubber that coats the wires in the phone here, the DNA wants to be at its lowest possible energy form of a certain number of bases per turn. And so to relieve this, you'll get like a kink forming in the phone cord. And we'll see that on the next slide. So with DNA, enzymes called topoisomerases can break the double-stranded, can break the strands. In this type 1 they break one strand, and type 2 they break both, but in either case they can add or remove turns from the DNA. So here no turns can be added or removed because the ends are blocked. With type 1 topoisomerase, if this is, so this isn't a circle, but here's, these two fingers are one strand, okay, and these two fingers are another strand, so here's two strands of DNA. Type 1 topoisomerase will break one strand and then turn it around like this and cause it to form again. You see that? So break it, turn it around, right? So you're either adding or removing one turn. Type 2 topoisomerases break both strands at once, pass the whole piece of DNA through, and close it up again. So because of that, topoisomerases are going to add or remove two turns. In either case, breaking and reforming the phosphodiester bonds and adding or removing turns will lead to, if you add turns, like if you turn it to the right, DNA is a right-handed helix, but now you've added two extra turns if it's a type 2 topoisomerase. And this little part of the DNA here is wound too tightly, so to speak. Or if you unwind it, then it's not wound tightly enough. The DNA, just like the phone cord, wants to go back to its certain number of turns or certain number of bases per turns, and so what will happen is this will unwind and this will rewind. But the ends are joined, so it's not like holding a phone cord up like this and letting it unwind by itself. What happens is you get supercoiling. So you get supercoiling in a direction that's opposite of the coiling. So like with the phone, you get some coils here, right? So two supercoils are added. So if it is, if you turn DNA two turns to the right, the supercoils are turns to the left. It's opposite, so it undoes it. But if you follow around here, the DNA now is all 10 base pairs per turn. There's no area here where there's too much. Likewise, if you follow this all the way around, there's no area where it's unwound here. It's all 10 base pairs per turn. But in this case, because you unwound the DNA, that's taking out two right-hand turns or adding two left-hand turns, you're going to add right-hand turns back here. In each case, you get supercoils, and you can see, look at this guy right here. Yep, see that? That takes up a lot less space than this. It's sort of all wound up tight, like if you took a rubber band and just twisted and twisted and twisted, it would take up a lot less space. So this serves two purposes. The first one I've already talked about is that it makes the DNA smaller, so you can package it into a smaller area. The second thing is, by adding negative supercoils like this, and your DNA and bacterial DNA is almost always kept in a negatively supercoiled form, you have the ability to go from this to this without breaking any phosphodiester bonds. So the energy for converting from this form to opening it up like that is very little. You're just removing the supercoils, and that causes the DNA to unwind. Why would you want to do this? Well, as we'll see in the next chapter, in chapter 13, you need to open up the DNA to single strands, both for DNA replication or copying of the DNA to occur, and also for making RNA copies during transcription. And so holding the DNA in this negatively supercoiled form means that it's easier to get the strands to separate, to break those hydrogen bonds that hold the DNA together there. and have that open up. All right, so like I said, if you want to unwind this, it's pretty easy to do. So this is the first level of compaction. We're going to talk about prokaryotes and eukaryotes. For prokaryotes, there's just a little bit more than this. For eukaryotes, we're going to talk about histones, but this is the first level here. So this is one of my cool slides, or one of the cool pictures in the book that I really like a lot. This is a bacterial cell that has been lysed very carefully. So it's been treated with detergent or something else that will allow the cell wall to burst, but not strong enough so that the whole cell explodes and goes away. And so all this stuff that you see around here in this electron micrograph is the chromosome of the bacterium. So there's a lot of DNA stuck in there. So DNA is negatively supercoiled, and then in prokaryotes, each of these twisted parts are then held together by proteins. And the proteins interact with components of the cell wall, so the peptidoglycan cell wall, and this is referred to as the nucleoid. The nucleus is in eukaryotes. It's membrane bound, and it's an organelle. Prokaryotes don't have organelles, but the old version of what a cell looks like, cytoplasm is just like a beach ball full of water and everything is sloshing around, is not accurate, not even for prokaryotes. In fact, what happens is more like the cytoplasm is a gel and everything is sort of in its place, and these proteins anchor the genetic material to the cell walls. We're going to take advantage of this coming up in lab in a few weeks. So next week I'm going to give you plasmid DNA. It's a small circle of DNA. You're going to transform it into cells. Then we're going to grow it up, and the next week you're going to take a mature culture of these cells and lyse them or split them open by using high pH. So we're going to add enough sodium hydroxide to bust the cells open. And we're going to want to collect just the plasmid DNA out of the cells, the little circles that are a few thousand base pairs, versus the E. coli genome, which is, I think, 5.8 million or 6 million, something like that. The way we get the plasmid DNA away from the genomic DNA is that we're going to precipitate out all the cellular proteins using a detergent called sodium dodecyl sulfate, or SDS. That is, if you look on all of your shampoo bottles, you'll probably see that as one of the ingredients. Sodium lauryl sulfate, sodium dodecyl sulfate, sodium laureth sulfate, they're all kind of the same things. So the detergent will precipitate out all the proteins, but because the DNA is connected to, the genomic DNA is connected to proteins, it will precipitate out too. So when I tell you to handle the tubes very carefully while we're doing this procedure and not shake them up or do anything like that, this is why, because we want to very gently handle it so that the genomic DNA doesn't rip away from the proteins, because if it does, we won't be able to collect it. All right. So eukaryotic DNA is a lot more complicated than that. As we'll see, that's a prokaryotes versus eukaryotes, more complexity is a theme. So level 1, naked DNA. This is if you take DNA out of a cell, you extract the DNA with phenol chloroform, which is a lovely smelling compound or mixture, and you get DNA that is away from all proteins. The double helix is about 2 nanometers wide on average. The first level of higher organization is going to occur when you wrap this DNA around a complex of proteins called histones. So each histone, not this one here, the purplish little beach ball there, each nucleosome core contains 8 different proteins. Histone, 2 each of histones 2A, 2B, 3, and 4. So H is histone, H2A is the histone 2A protein, histone 2B protein, histone 3 protein, and histone 4 protein. The nucleosome is also sometimes called the octamer because there's 8 parts to it. What's shown here isn't completely what I would consider accurate. They always show the histones in this book as sort of like a beach ball. Really, you can think of the histones as more of like an elongated disc. So if you think of a spool of thread, but if you crunch down the spool so it's not, it's a very, think of it as a shortened out cylinder, not a beach ball like this. In any case, the DNA is going to wrap around the histone core. We're going to come back to the last histone, H1, also called the linker histone. That's going to come in later. They show it together here, but really it's adding this histone that gets you up to the higher level of organization, which we're not talking about yet. All right, so about 150 bases of DNA wraps around each histone. It wraps around almost 2 times, but not quite. And then the DNA between each histone is called the linker DNA. This is longer at this point than it will be later because the histone H1 is going to shorten that, but it's not there yet. So now we're going from a 2 nanometer fiber, which is the naked DNA, to what I refer to as the 10 nanometer fiber. The book says 11, everybody else says 10. So if you put 11, I'm not going to take off for it, but the 10 nanometer fiber is what everybody else says. So this is sometimes referred to as beads on a string. So here is 2 electron micrographs. If you look here, the little dots are the histones, and so there's DNA wrapped around them, and the string is that you can see between them is the linker DNA. Here it is a little bit more closely. Here's one histone, linker DNA, another one. Like here's one histone, linker DNA, another one, and it just goes on and on and on. So you can go from the 2 nanometer fiber to the 10 nanometer fiber, and by wrapping around the histones, you're compacting the DNA. Actually, wrapping around histones is another kind of supercoiling, right? And you guys have all, hopefully, all experienced this. How many of you guys have tried to like coil up a garden hose, right? You like wrap it up into a nice circle, and as you're making it into a circle, just like this goes in a circle around the histone, you're adding twists to the garden hose, aren't you? Because you have to keep throwing the end and spinning it around like this, or it all gets into a huge mess. So wrapping around the histones is another form of supercoiling that occurs. The next level of organization allows you to go from the 10 nanometer fiber shown here to a 30 nanometer fiber. But first, let's have a look at this. So this is going to be a better view of what's going on. So down here, entering at the bottom and then going around 1 and almost 2 times, coming out up here, is the DNA. So if you can imagine the cylinder of the histone is coming out towards you, so it's sort of like this, right? Coming out towards you like that, and the DNA is wrapping around it like this, going towards you. All of these colored strands in the middle here are the turns of the helices of the histone proteins. So the red and yellow are histones H2A and H2B, and the purple and green are the histones H3 and H4, and so together they make that octamer protein. What? Oh, the H2A and H2B are the red and yellow. H3 and H4 are the purple and green. I'm not going to ask you to identify which is which on this picture. This is an extra thing that I got out of the molecular biology book, because instead of just looking at a little blob like this is the actual molecular structure of what DNA wrapped around a histone looks like from a crystal structure, to help you see it more easily. So, yes? All right. So, the core nucleosome is also called an octamer because it's got 8 parts to it. What? Yeah, same thing. So, this is the core, and then the complete nucleosome also will include the linker histone H1, which we're going to get to, and I don't like that they show it in this figure, because in the 10 nanometer fiber, the H1 shouldn't be there yet, because adding the H1 would make it look like something else that I'll show you in a second. All right, so how does this interaction occur? Histones have to be able to interact with DNA regardless of the base pair sequence. It has to be non-specific interaction. And so histone proteins have positive charge to them. That allows them to interact with and form hydrogen bonds with the negatively charged, there's ionic interaction and hydrogen bond interaction between the histone proteins and the octamer here and the sugar phosphate backbone of the DNA. So histones are positively charged, the phosphates are negatively charged, and it kind of holds it together there. This also allows the DNA to come to be moved from one place to another on a histone. So important for because in the next slide it's kind of nondescript, but look, see this little tail coming out here, and this one, all these have these little tails that match the color of them coming out at the end there. See all those, right? These are the histone tails at the N-terminus, or the amino terminus, if you're thinking about proteins. And these are going to be very important for how tightly the histone is binding to the DNA. Remember we talked about like open and closed chromatin, so the DNA is more accessible or less accessible to proteins. You get some interaction here, but these histone tails are going to cause something else to happen that I'll show you here. All right, so here is the book's version of that, and you see the histone tails coming out of the nucleosomes. Now they're not extra, they're part of all the proteins, so there's going to be 8 tails coming out of there. Under normal circumstances, these tails will be positively charged, just like the histones. So they will be latched on and gripping the DNA tightly. I'm not so enamored of this picture that they've put in for the 6th edition of the book, because the tails are just sort of floating out and it doesn't look like they're grabbing the DNA at all. But these should be like reaching around and gripping onto the DNA at this point, right? That means that the histones are interacting very tightly. The chromatin would be in a more closed conformation. All the histones, so yeah, H2A, H2B, H3, H4, all 4 kinds of histone proteins have the tails. So, under normal circumstances, the histone tails are positively charged, and so grip the DNA very tightly. However, enzymes called histone acetylases or histone acetyltransferases, HATs, will add acetyl groups to the N-terminal tails. Right? Acetyl group, carbonyl, carbonyl with a negatively charged oxygen hanging off the end of it is negatively charged. So these negative charges counteract the positive charges, and this is what makes the histone tails, which should be shown up here as tightly gripping the DNA, go down to not be gripping the DNA and be more loose interaction. So up here, the DNA would be tightly gripped and maybe not so accessible to proteins that are going to do transcription or replication. Down here, it's a more open area. So areas in the genome where transcription is actively happening are probably going to be in this open conformation. Areas in the genome where transcription is not actively happening are probably going to be up here in this closed conformation where they should have drawn the histone tails grabbing the DNA more tightly. So in addition to histone acetyltransferases, which are going to add acetyl groups, the opposite can happen, and that's shown here. So deacetylation can be done by histone deacetylases, or HDACs. They will remove the acetyl groups and therefore cause the N-terminal tails to resume their positive charge, grabbing the DNA more tightly, more closed chromatin conformation. And this is not a all over the genome phenomenon. It's specific to local areas, and it can be changed. As we'll see in chapter, I think 16, the eukaryotic transcription regulation specific chapter, there are also other modifications that can be put on these N-terminal tails that form a more specific code. So global, adding acetyl groups to make them less positively charged, or removing the acetyl groups to make them more positively charged, that is sort of like the big sledgehammer. But there can be the fine-tuning by adding specific modifications to specific parts of each of those histone tails. Don't write it down now, but the modifications are going to be, there's acetyl groups like this, phosphates, and methyl groups. And so the combination of all those different modifications to different parts of the genome are going to make out a code that can say a little bit more nuanced what should happen at that area. All right, so 10 nanometer fiber. So naked DNA is 2 nanometers. Wrap it around histones, that's beads on a string, 10 nanometer fiber. If you add histone H1, what happens, and it's not shown on this unfortunately, the histone H1 will make more of the linker DNA be grabbed onto by the histone octamer. It shortens the distance between the histone octamers, and that makes the DNA go from being beads on a string like this. So here's the DNA wrapping around the histones, and then the DNA wrapping around the histones is wrapped into what's called a solenoid, or a 30 nanometer fiber like this. Do you see what I'm doing with my finger here, like going around like that direction? So you go from this is a 10 nanometer fiber to this thing is 30 nanometers, and that's what this looks like. So you can do this in vitro. You can take naked DNA and add the histone H2A, H2B, H3, H4. They will spontaneously form into this beads on a string, 10 nanometer fiber. If you add to the test tube histone H1, that will shrink the distance, that will shrink the amount of DNA between the histones, and you will immediately snap from the 10 nanometer fiber to this 30 nanometer fiber. And these are both electron micrograph pictures there. The way that this happens is, so this should not have histone H1, but if you take the 10 nanometer fiber and add the histone H1 will interact with both the the octamer there and some of the DNA that formerly was not touching the nucleosome, but now it sort of winds it in and makes it go closer together so that you go from this 10 nanometer to the 30 nanometer fiber. That's all I'm going to ask you about. I'm showing the rest, but I see no point in making you memorize these numbers. So don't worry about that. But it's, yes? So the 2 nanometer fiber goes to the 10 nanometer fiber. Histone H1 should not be there. Yeah, so somewhere down here, I mean, it's not, you're not doing it exactly like this. You're going to add the histones and you're going to add the, right? It's never looks like this. This is just a representation of it. So if you add the so here, the 30 nanometer fiber is this solenoid structure. If you were able to magically remove the histones, like if you had something that degraded the histone H1 specifically, but left the octamer histones alone, or like an antibody that bound to histone H1 and removed it from the situation, you would go from the 30 nanometer back to the 10 nanometer. So they're showing these here, but adding these makes it go from the 10 nanometer up to the 30 nanometer. That winding it more tightly, that's what makes it want to go into that structure. And then you have interactions with other proteins that eventually get you up to the chromosome structures that you see at metaphase. I showed the bacterial cell lysed with the DNA all around it. This is like another one of my favorite pictures here. So remember I said that DNA, or that chromatin, which is DNA plus proteins as it exists in a nucleus, is half DNA and half protein by weight. That's why scientists were unsure whether protein or DNA was a genetic material. That plus protein is more complicated than nucleic acid as far as structure. So this is a chromosome like that was very gently digested with nucleases so that the DNA has been digested away, and you can see this X here. That is the protein scaffold that makes up a metaphase chromosome. All this other stuff, which you can only, here's the edge of it, but you can't see the top left or right because there's so doggone much of it, that is the DNA that came off that chromosome. So they treated this very lightly with nuclease, enough to get the DNA off the chromosome, but not enough to digest it completely into nucleotides. So you can see the astounding level of compaction that happens when you put that much DNA onto one of these chromosomes that are required for doing meiosis and mitosis correctly. Because this is easy for microtubules to connect to the kinetochore proteins here that bind to the centromere sequence, but you try to take this much DNA and pull it to one end of the cell. It's like a never-ending, it's like a bowl of spaghetti where each strand of spaghetti is super long. You're just never gonna get each chromosome completely to one side of the cell because there's too much of it hanging around there. All right, so in eukaryotes, which of the following would you expect to be most tightly coiled versus least tightly coiled? Think most tightly coiled is going to be inaccessible, and least tightly coiled is going to be more accessible to proteins. Which of these would be, so which of these would have to be loose and not tightly coiled in order for it to work? Yeah, the two at the top, right? Actively transcribed DNA, you have to be able to open it up. So proteins have to be able to access the DNA, and then the DNA has to be able to unwind in order for you to make an RNA copy. Same thing for DNA that's replicating. The only difference between these two is when the two strands of DNA separate to make an RNA copy, they will come back together because you're just making an RNA copy and it goes away. When DNA is replicating, on the other hand, the two strands come apart and a second strand is added to each of them and they stay apart. DNA that codes for proteins not expressed in a particular cell type, in fact, that should be kept as very tightly coiled DNA. The reason being that you don't want proteins to be able to access the promoter for that. You can show this accessibility versus inaccessibility with an example like this one that where you're talking about genes that are expressed at differential times during development. So globin genes encode the globin that is part of the hemoglobin that's required to transport oxygen through your body. So this is what's going to be in red blood cells. Right? So erythroblasts are red blood cells. During the first 24 hours of development, you get no hypersensitivity. So what is this? There's an enzyme called DNase that I talked about for the previous slide that will cut DNA molecules. In this case, it cuts them non-specifically. So you can do an experiment that I don't have time to go through here, where you treat them with just a little bit of DNA and you can see where in a certain piece of DNA is going to be the most sensitive or the most likely for these cuts to occur. You put enough DNase in and eventually you're going to chop the DNA up into little pieces. At 5 days, the gene for the U form of globin has at its promoter region an DNA hypersensitivity site here. So that means that DNase is able to reach and cut the DNA here, but not at these two spots. These two genes are turned off, this one is turned on. It makes sense, right? So the area that's accessible to proteins that are required to turn the genes on for transcription, that also means that same DNA is going to be open to the bad news enzyme that is DNase 1 to chop it up. Then you get a switching of which genes are coding for globin as the mouse goes through development. So sometime between 5 days and 14 days, you go from the immature form of globin that's from the U gene to the mature form that's found in adult organisms, the alpha globin, and there's two different, the D and the A versions of that, two separate genes. So now you get the ability of DNase to cut the DNA here and here, but not over there, because you've switched which genes are transcribed. So yes, you require transcription factors to make an RNA copy of the U, the alpha D and the alpha A genes, but up here, even if the transcription factors that would turn on the alpha D and alpha A genes were present, that DNA is in such a tightly closed conformation that no proteins can reach it. If the DNase enzyme can't reach these regions to cut them, then the transcription factors can't reach it either. Versus this is just the opposite. Now the area around the promoter of the U gene for globin has been restricted and these two are open. So you get a switching in chromatin conformation. As a control, you could look at brain cells or pretty much any other cell type besides erythroblasts. No other cell has a reason to turn on globin genes, and so these genes throughout development are in a tightly compacted form of DNA. So this ensures that they're turned off, And even if the transcription factors that would normally turn on these promoters are present, they still wouldn't be turned on. That allows you to use the same transcription factor for multiple genes. The transcription factors that normally turn these on may be turning on some other genes in the brain, but because these are closed down, we don't have to worry about them being transcribed. First, we don't need globin in neural tissue. And second, so it would be a waste to make it. And second, in a lot of cases, making certain proteins by transcribing the genes into RNA and then translating them would be harmful to that cell type. And so you want to very tightly control that. So this is an example of not only in a specific cell type, so all three of these genes are only ever expressed in red blood cells. However, these are expressed at different times, during development there. Okay, so we're going to stop there. I'll see you guys during the lab if you have the lab today. Otherwise, I'll see you on Thursday.